So after you have selected the top differentially expressed genes from your data set, then we can do gene set analysis to try to make sense out of it, especially if you don't know much of the biology of the system that you're studying. There are many ways of finding what is the function of those genes or the cell type of those genes are related to. And then we will see that also in the cell type prediction lab later on as well. So here I'll do a brief overview of gene set analysis. And this is, if you have already done RNA-seq before, this is the same thing, or proteomics. What we do for single cell is just take that list of genes and try to find sets that are related to a certain function. So imagine that you have the genes that are up or down regulated in a, I don't know if you can see better if you have a laser point. So the genes that are up or down regulated in one condition, and then you want to find their function, which means you can associate them to gene ontology terms that I'll explain a little bit more, to pathways, or maybe to cell type, maybe that cell type is enriched, chromosome locations, transcriptor factors, histone modifications, diseases, and so on and so forth. So the main idea of gene set analysis is transform these gene level data, up or down regulated genes, to up or down regulated pathways. So then you get an overview of what is happening in your data. In terms of single cell, then you want to find, oh, this pathway is upregulated in the disease versus control in that specific cluster. So here, most of it is transcriptomics and differential gene expression analysis, but you can use it for anything else. Even if you have ATAC-seq data or you have methylation data, spatial transcriptomics, anything is valid. So gene set analysis is an interpretation of these transcript-wide results. And the gene sets are usually fewer than the genes, so it's much easier to have an interpretation. Instead of 20,000 genes, you can set up to 400 pathways, and of course, not all of them will be significant. And of course, it's really hard to find like significant genes per se. So if you have patterns, for example, like this whole pathway is upregulated, so you have a list of genes, let's say 100 genes that belong to that pathway, that gives you a more robust result that pathway is actually upregulated, rather than saying that gene from that pathway is up. So this is a better way to explain your data and have meaningful results. So in some cases, the top genes might not be the most interesting ones. So even if you look in the top of the list, so maybe ribosomal genes or mitochondrial genes usually come up. But if you just take a look at the other genes or the collection of genes that belong to your differential expression table, then you can have more significant or coordinated small changes, and that might be significant. And this is a way to integrate external information into your analysis. So in this case, pathway or function to the gene, but you can associate anything to that. And one of the examples, especially for single cell, would be to cell type identification. And that can also be done via GSE, and you'll see that on Friday, I think. And then that's also less prone to false positives compared to the gene level. So it gives you a more robust analysis, because you're not counting on only a single gene, but rather you're looking at the whole transcript landscape for that pathway. And if all those genes are upregulated, this pathway will be enriched.
May I ask something?
Yes, absolutely.
So for a bulk RNA-seq, you would, for example, often take a fold change cutoff for this type of analysis. Would you recommend to do that here, or would you maybe just take all differentially expressed genes?
That depends on the method. So for taking the differentially expressed genes, you need to have a cutoff, both p-value and FDR, both p-value and log FC. But that is used for the hypergeometric test that I'll talk soon. And then there is another method that you don't have any filtering, you just put everything into the method. I think I will answer your question in a couple of slides, I think. regarding like which method you can use and how you can filter that. Yeah. So the different gene sets will also depend on your research questions. So there are different databases that you can look for it. So the molecular signature database from Harvard, or the Enricher pathway, or the Panther, they are quite good tools or websites that you can look. And there are many tools already in those websites, but I would focus on the gene lists. Gene ontology, most of you are already familiar with it, are the most widely used, but there are, as I mentioned, different ways that you can interpret your data.